Good afternoon, folks. I've got 12:28 on my clock in the office, and now we have 1 minute left according to my clock in the office. So if you're present, type your name in so I can keep an accurate record of your attendance. Got to do that for the financial aid situation. I'm going to shut my door so hopefully nobody interrupts us. Hi, Shaniqua. Good to see you. Again, Sonia, did you hope you got something out of that other video? We're going to do the same thing today. Hopefully it won't shut off like it did this last video. Several kids said it just disappeared after I held the book up. So, oh, there's the real goddess. Hi, Kira. Kira. Hello, Anna Grace. Got to stop doing that. I tell you to keep your hands away from your nose and your mouth. I've got to break that habit. I'm going to rub my nose. Hey, Madison, how are you? Cameron, how are you doing? Been kind of frustrating to respond to us, hasn't it? Good, Madison. Glad you're doing well. Kind of a trying time in the last week or so, isn't it? Hi, Tamika. Looks like you go from one screen to another. I see Mr. Davis has got a little bit of a conference scheduled with you. That's good. Hey, Morgan. Hey, Destiny. Good to hear from you. You guys can go ahead and turn to page 345. Going to have just a bit of a review, and then we'll move on. Yeah, we're going to start on in just a minute. We're going to be behind. Well, Courtney finally here. And Caitlin, good. Got to meet Mr. Davis after this, don't you? Hey, Elizabeth, finally. Glad you're here, Elizabeth. Hope it wasn't too much of a problem. Hey, Madison. Destiny, we're going to be on about page 345. It's that big picture type thing up at the top. It's figure 10-4. Hi, Jason. Jason, you haven't heard anything about Rodney, have you? Seems to have disappeared. Thanks, Jason. Appreciate that. Hope you hadn't gotten sick. Hello, Mason. Good. Okay, well, I've got 1240, so we're going to start. So you should be on page 345. Page 345. We covered most of that. I just want to go over a little bit of a review with you, get you thinking about this muscle cell. So as you look on page 345, figure 10.6 has the big picture of levels of organization within a skeletal muscle. And you come to the middle. Hey, Stephanie, good. James. Thank you, Stephanie. So you already know the structures up top, the tendon, the epimysium, the fascicle, which is a collection of muscle fibers. When you look at one of the muscle fibers in the middle, and you see it's labeled a muscle fiber, what's another name for a muscle fiber? It can have three names. What's another name for a muscle fiber? Good. Very good, Courtney. Very good, Elizabeth. Muscle cell, myocyte. Mm-hmm. Good. So as you look at this muscle fiber, let's get a good look at it. Got several things I want to ask you about it because we're going to move into how contraction takes place. Now, you see the sarcolemma, and you see these look like little logs that are inside the cell because that middle section is the picture of, or the illustration, I should say, of a muscle cell or a muscle fiber. And inside it, you see these log-shaped structures. One of them extends past the cut face. And what did we call that structure that looks like a log? Getting you to think about this because this is, it's kind of deep, and you're going to probably get the test next week. By the way, speaking of the test, you don't have to worry about Respondus anymore because it is so selective in what computer, laptop, or whatever it works in. So they have gone with HonorLock, and that allows you to do it on your personal computer. It allows you to do it on Chromebook. It allows you to do it on Mac computers. It allows you to do it on tablets. So it looks like it's kind of an open door. That's the way they want to go with HonorLock. Now, how do we get it set up? Hopefully they'll tell us that sometime during this week, and I'll get that set up in D2L. Okay, back to okay, good. I see there myofibril, and Madison, you're correct too. Myofibril. Now, we said that a myofibril had another common name, which could include a number of structures. Do you remember what the myofibril, which is only found in muscle cells, what they would call that structure that is found inside the cell? Common name includes a lot of other structures. Organelle, that's correct, Courtney. So we've got a number of organelles in the muscle cell. As you look at this picture, I'm not sure exactly how accurate it is, but you can count the myofibril. That's right, Mason. You can count the myofibrils. In here, you got 1, 2, maybe 3 over there past the one that's extending out. 4, 5, 6, 7, 8, 9, 10, 11, 12. That's showing about 12 myofibrils per muscle cell. And I was looking at some stuff last Monday night, I guess it was, maybe Sunday night, and they mentioned something like 6 myofibrils per muscle cell. But the point is, you've got a number of those myofibrils in a muscle cell, and they are organelles. Name some other organelles that you would expect to see in a muscle cell. Name some other organelles that you would expect to see in a muscle fiber. Good, Courtney. Mitochondria, boy, all of you are right nailing on that one. Good. Mitochondria is one. What's another one that you'd find in another cell, or even in this cell? Good, Tamika. Golgi apparatus. Good, Destiny. A nucleus. Anything else in there? Good. Ribosomes. They're going to play a role in there. That's right. Rough ER. Very good. Sonia, you're right. Mitochondria. Got the rough ER. Got the smooth ER. Don't forget that you can have, that's right, Shaniqua, the Golgi apparatus. And don't forget those little vesicles like lysosomes. Okay. That's right, Sonia. Good. Okay, so you got the picture again. So as you look at this picture, this middle picture, you see they've got 12 myofibrils in that cell. So there's some difference of opinion apparently, because I only saw somebody say there were 6 in there. Now let's take that reddish area. You see the myofibril that's sticking out, and you want to follow that kind of reddish area down, and you got a piece of that organelle. You have a piece of the myofibril, and if you look to the left, you see a light area that has a little zigzag line that runs down through the middle of that light area. You also have a light area over on the right-hand side with the zigzagging line going down through there. That light area is called what? It's not a T-tubule. Jason, you're right. Madison Becker, you're right. I band. Those 2 light areas that I just mentioned, they are the light band. So the band in the middle between those light ones would be called what? That's correct, Destiny. The I band. So what would the darker area, the redder area, be named? That's correct, Madison. The A band. And of course, those bands are the result of the protein filaments. Now, as you look at that little Z line, that little zigzagging line, which they sometimes call the Z line, hang on to that H-stone, Destiny. We'll probably use it in just a few minutes. So as you see that Z line, sometimes they call it a Z disc. and then you look at the other one at the other end of this myofibril, that section of or collection of proteins in that area, that section has a particular name. And what is that section called? Between the Z lines. Okay, Madison Becker, you got it. That whole structure that's got half of a light band on either end and then got the darker band in the middle, that's called a sarcomere. Now, if someone were to ask you, that's correct, Stephanie, good. Madison Oaks, good. Madison Oaks, if someone asked you what in the world is a sarcomere, what would you say to them? How would you define that structure? Good job, Shamika. It is that name, a sarcomere, but how would you define it? How would you explain it? What's its purpose? Okay, Kira, that's right. They probably would spell contractor differently. Contractor is somebody who's going to build a house for you, a contractor. But if you take the OR off and put an ILE on the end of that T, you have contractile, and so you could call it a contractile unit. Courtney, it is a structural unit, but contractile tells you it's going to be involved in the contraction of the muscle. That's correct, Madison Becker. The space in between Z line to Z line, that is a sarcomere, which is the contractile unit. That's right, Shaniqua. That's the function of it. It is the contractile unit. Now, as I was reading the other night, Sunday night, and they were talking about a biceps, and of course, you got a lot of muscle fibers in the biceps. And they were saying that the biceps had something like, and I think I've got this correct, I'll have to go back and look at it again. They had something like 80,000 to 100,000 sarcomeres in that muscle. That's a lot, isn't it? 80,000 to 100,000 sarcomeres in the biceps muscle. So obviously you'd have fewer ones in smaller muscles, like the little muscles that allow you to flex your fingers like this or squeeze your hand or whatever. So got a lot of contractile units, don't we? Now let's look down again. Come down from that myofibril, and you look at that bottom diagram, and you see the title of that is myofilaments. It's a magnified version of what we were just looking at, from one Z line to another Z line, from one zigzag line on the right-hand side to another one on the left-hand side. So now you've got it enlarged, and we actually have, let's see, 1, 2, 3, 4, 5. Yeah, I think that's right. 5 different proteins presented in that illustration. One of those, if you look at the Z line, we're looking at the bottom illustration. If you look at that Z line that's zigzagging, that is a type of protein that they call the Z line or the Z disc. It anchors another protein, which is the little skinny purple one. You see 3 of those. That's actin. actin. That's the actin protein. You also see the red, bushy looking, kind of looks like it's got little heads on it or balls on it. That's your myosin, myosin. That's a different protein. So we've labeled 1, 2, 3 different proteins. And notice, look at the myosin, look at the tip of the myosin, and you see this little coil. Now, if you look over to the left, on that diagram, you'll see elastic filament. It looks like a little spring, doesn't it? So that's another protein. So we're up to 4 proteins just looking at that. We got a few more to go. And then you look in the very center and you see 3 lines, don't you? That's the M line. That is another protein that anchors the myosin into position. So there you have 5 proteins, and we got a couple more to go in just a few minutes. But that's just 5 proteins. Now think about the difference between them. What makes actin actin and myosin myosin? And that little elastic filament is called titin, Titin. I think we mentioned that last week. Titin. It looks like a spring. See, look at the different shapes of those proteins. Remember, the amino acids, the number of amino acids, the sequence of the amino acids, that all determines the polypeptide, which is going to turn into some sort of a protein that we can use in a specific way. Causes them to be in different shapes, but those shapes dictate the function. Just like a hammer. A hammer has a shape. Most of you know what a hammer is, but you can't do with a hammer what you can with a screwdriver. Different shapes carry out different responsibilities. The same thing is true in the muscle cell. So you can see the seriousness of having the right amino acids in the right sequence, the right number of them, so that you get a particular shape after you go through the primary. Remember the primary structure? Then you got the secondary structure, tertiary structure, quaternary structure, and that all occurs inside the ER, so that a particular shaped protein comes out that will carry out a responsibility in our bodies. Just mind-boggling to me. It's fascinating engineering. It's just wonderful. Now, as you look at that picture, you can see how you have sort of an overlapping or a space between, I guess you'd say, some of the proteins. Like you start at the top where you see actin, the little skinny one, then you got a myosin, then you got an actin, then you got a myosin, then you got an actin, and so forth. So they line up like this. And they're sitting there. Some of your muscles are sitting just like this. As you take notes, as you shift your weight in the chair or whatever, what you do is you make these muscles do this. That's contraction. The actin is pulled in toward the center of the myosin. Now, we're going to get into that in just a minute. Just trying to get you ready to think about that. So, let's look over now on page 344, figure 10.5. And as you look down at the bottom, you see myosin. You see myosin is made up of a number of skinny little tails and a bulbous head. That's just one part of that protein. You can see you got a bunch of them there together. There must be 25 or 30 heads on each end, and you've got those little tails in the middle, probably somewhat hooked to the M line, which is a different protein and holds those guys in the right position. You don't have to think about that. That's just the way you're built. Now, I want you to come down to the little, looks like a bunch of purple grapes. They're sort of rotating like a little coil or a spiral. And you see over here on the right-hand side, below you see the word actin. And so this is the protein actin. This is one of the units, the little ball, the little grape-like structure. The name of the figure, Destiny, that we're looking at is called structure of myofilaments. 10.5. Should be on page 344, unless you got an older book. Anybody got an older book where it's on a different page for Destiny? So we're looking at myofilaments. Now remember, I said that most people, when you think of myofilaments, you think of the actin and the myosin. But there are other proteins involved with that. If you don't have the other proteins, got a problem. Big problem. You're on the e-text. Whoa. I don't have an answer for you there, kid. Katie says go to page 344 if you can find that in your on your computer on the e-text. I don't have that drawn up at this point. You could see the big picture though, couldn't you? Have you gotten it yet, Destiny? Come on. Let's see here. Huh. I can't even get past it. You see it now? Okay, good. I can't even open that up on my. How about that? Better not X that out. I might be having a problem with that. Anyway, okay, you got it. So you see the myosin tails, you see the myosin heads, and you see what that one molecule of myosin is composed of, that one tail and head. You got a bunch of tails and heads. So you look at actin down below and you see the purple, look like grapes, and that's actin. Now come down to the last little bit where you see the purple grape all by itself, and you see they say that's an actin subunit. And then you see the little hole in that subunit. That's the active site. That's the place where the head of the head of myosin attaches to actin in order to move it, in order to have contraction. Now, we've got to look at a couple more proteins. So as you look at that twisting purple strand of grapes there, which is really actin, look at on the right, you see this word called tropomyosin. That's another myofilament. It's another protein, very thin protein. And tropomyosin covers the active site of actin. So if tropomyosin covers the actin site, myosin cannot make a connection and cause a contraction. Therefore, your muscle is relaxed. So that tropomyosin maintains a state of relaxation in terms of muscle contraction. One more protein. You look to the left of tropomyosin and you see troponin. Now, troponin is represented by 3 little yellow balls. And you see they got a couple of those molecules of troponin. Troponin is the protein that moves tropomyosin. It moves tropomyosin to expose the active site of actin. James, how you doing? Let's see, where was I now? So when contraction occurs, and we're going to go through that a little bit, troponin is the one that makes tropomyosin move. So the actin site, the actin active site, sounds real clumsy in your mouth, doesn't it? The actin active site is exposed, and the head of that myosin fits in there, makes a connection, and pulls the actin in toward the M line. Now, before we leave this particular page, look to your left and you see that spiral protein called titin. That looks like a spring, doesn't it? Okay. Courtney, you needed me to repeat that about troponin. Whenever you're going to contract your muscles, and we're going to talk about how that happens in a minute, but troponin changes the shape of tropomyosin. It's going to move it so that the actin active site can be connected to the head of myosin. Is that better? Good, good. It's amazing to me to look at that and think about how that looks so, I'm lost at a word, whatever it is, graceful. That seems so ungraceful. And yet you look at somebody, some athlete like Simone Biles, who can do all that work with her body, and she's like fluid in motion. And that's what's happening. The head of that myosin is hooking up to the active side of the actin, and the head pulls the actin in so that contraction takes place at just the right time. That's why I'm teaching this stuff. I'm amazed at it and I love it. All right, so much for that. Let's look to the left, and you see that coil. They call it an elastic filament. It's a protein. It's called titin. 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 Now, some of you will know this. Some of you have no idea that this is the case with a car. With each wheel that you've got, it has a tire on it. There is an arm that the tire is hooked to on either side. And when you hit a bump, this happens. You hit a bump on one side, that tire kicks up a little bit, and then it comes back down to its regular position. This protein filament called titin, and look at the picture that has the rectangle, and you see that coil. It's hooked to the myosin and it's hooked to the Z line, isn't it? It's two places where it's hooked. Just like there's a couple of places for your suspension in your car where there's a spring. So when you contract your muscles and these actin filaments slide over the myosin, what happens whenever you relax? Got to have something to push them back. And the spring, that titin, pushes them back. Just like your springs push that suspension arm and that tire back down to the road so you have a nice even surface. I mean, even four tires on the face of the highway. You go over some rough road, your tires will do this number. They're changing because of the unevenness of the road. But the spring always brings that tire back down to the road so you're in good contact. Shock absorbers are another thing kind of like this spring. So it goes up and it pushes them back down. So this one, in this case, the spring would push these fibers back a little bit, these filaments back, and you would be relaxed. Okay? Comfortable with that. Let's look on page 348. And what you're looking for, Destiny, is figure 10.9. 10.9. And you see it says, for a title down there, it says the sliding filament mechanism, which is a little bit of what we just discussed. So look at the top of A. You see the I band, and there's your H zone that you were talking about, Destiny. That's on either side of the M line. That's the H zone. But you see up top of the illustration, it says relaxed sarcomere. Now notice the position of the actin filaments. Those little skinny, kind of light purple marks, not much bigger than a 0.5 pencil lead. But you see their position. So whenever contraction takes place, look at the bottom half, and you see that actin on either side has been pulled toward the M line. And what happens is you shorten the sarcomere. That's contraction. Anytime you have contraction, the muscle will get shorter. And then when you relax, it goes to be like it was up there at the top, where there's a greater space between the actin and the myosin filaments. That little spring has pushed things back, and the muscle has relaxed. So that gives you a picture of what's happening. Let's go over to, in my book, it's 352. Page 352, and you see in the first column, it says 10.4, the process of skeletal muscle contraction and relaxation. Does everybody see that? Anybody out there still breathing? Somebody give me a yes or no. Oh, good, good. The reason I asked that is because this morning we were just about to the end and something started, they said it was buffering or whatever, and we lost the video. Okay, Katie Hurst, what year are we on? I want you to look at figure 1012. At least that's what's in my book. 1012. Thank you, Tamika. Thank you, Madison. Both of you Madisons. Should I talk more slowly? I'd be glad to do that. Got no problem doing it. I want to make sure you get this. Got it, Katie? Okay. Now notice in that first column, we're under the heading of the process of skeletal muscle contraction and relaxation. Come down that column and you see this title called the neuromuscular junction. And obviously, you know, neuro means nerve and muscular means muscle. You can also call it the myoneural junction. Myo meaning muscle, M-Y-O, and the neural, N-E-U-R-A-L. So either of those terms is satisfactory. Myoneural junction, neuromuscular junction. It's nice when they name things and you know exactly what they're referring to. The figure name that we're looking at, Destiny, is structures of the neuromuscular junction. Structures of the neuromuscular junction. We haven't generated that. You might be looking at a different thing, Mason, different picture, generation of the membrane potential. Yeah, structures of the neuromuscular junction. That's what we're looking for. Structures of the neuromuscular junction. That's the title of the image. Did you see what Anna Grace said there, Destiny? It says it's figure 1014 in the e-text. Okay, good. I thought you would think that publisher would put all this stuff together, wouldn't you? That's because humans are doing it, and we don't always do the most efficient job, even though we might try. 353 in the e-text. Okay, you got it now? Great. Okay. Everybody's on the same page. So as you look to the right, you see the illustration or the image, don't you? And look above and you see a muscle cell like you saw before. The only thing different is you don't have one of the myofibrils. Good girls, good. Now look at the motor neuron. That's the nerve that's coming down. And then you see it sends branches to the muscle fiber. Now what I want you to do is you look at that little, it's almost like a little camel foot. You got 3 of them there, don't you? Sort of yellow. Look where the one's got, for the camel foot or that little yellow structure, we're going to mention it in just a moment, has that box around it. Come on down with that blue arrow. And we're looking at an enlarged version of that. So you see that little axon terminal? That's that bulbous sort of yellow structure. And you look down, you see on the left-hand side, it says axon terminal. That means the end of it. You see synaptic vesicle. We're going to talk about that in just a second. And within the vesicle, you see acetylcholine molecules. Acetylcholine is what we call a neurotransmitter. A neurotransmitter. So it's going to carry messages from one part of the system to another part. In this case, it's going to be from the nervous system to the muscles. So you see the synaptic vesicles inside, and you see the acetylcholine molecules inside the vesicles. Still on the left-hand side of that illustration, come down to where you see sarcolemma. Left-hand side, come down to the word sarcolemma. Now, you know that is the cell membrane, a phospholipid bilayer, right? And you see how it dips down and curves around like this. And you see it's got these little purple structures on it. Now remember, this is a cell membrane. We know it's a phospholipid bilayer, but we also have proteins that transverse. They go across from one side to the other side of the plasma membrane. We looked at that in chapter 3. See, chapter 3 is going to keep coming up. It's got all these parts in it because we looked at the cell, and now we're looking at collections of cells from various parts of the body. So you see it says ACH receptor. That's a protein that penetrates the phospholipid bilayer. It goes from one side all the way through. So it goes from the outside to the inside. Cytosol is on the inside. Now, if you would, go to the third little protein, purple protein, and look right above it, and you see the word called synaptic cleft. Synaptic cleft. And we don't talk much about cleft unless you talk about a cleft palate There's an opening up there in the roof of the mouth that shouldn't be there. So when you hear cleft, you think of a space. You know, if you go read the book of Exodus, you'll find Moses wanted to see God, and God said, "No, you can't see me. Nobody can see me and live." And so, but God passed by him, and Scripture says he put his hand over him and he pushed him into a cleft of the rock. So Moses was hidden in the cleft of a rock while God passed by him. So it's an opening, just like this. It's a space between the axon terminal and the sarcolemma of the muscle cell. So since you've got a nerve joining muscle, you can call it the neuromuscular junction. Sometimes they'll use the word synapse, a junction between the nerve in this case and the muscle. Okay, everybody got the pictures? Now again, just thinking a little bit, trying to get you to think, consider those proteins that pass through top and bottom, outside, inside of the sarcolemma. What's the difference between those proteins and the proteins that are involved in contraction? Well, again, you got the types of amino acids, you got the number of amino acids, you got the arrangement of the amino acids. It goes on and on and on, just like our words that draw a picture in our mind, or we hear them and we understand what's being said. We can see the words and we understand. That's how we communicate. Well, this is communication here. Specific proteins for specific jobs. Thousands of different kinds of proteins. How did they get made? What are the two processes that end up producing proteins? Quickly, somebody write those two processes down. You might be asked them next week. The synaptic cleft is a space, that's right. But how do these proteins get made? These proteins that pass through the membranes. How did the proteins get made that were actin and myosin and troponin and tropomyosin? What do you want me to repeat, Stephanie? That's right, protein synthesis. What two steps are involved in protein synthesis? They start with T's. That's right, Destiny. What are the two processes involved in protein synthesis? You see how many questions I can get out of this stuff? That's why you got to know it like the back of your hand. Anyway, you can look it up. Okay, half right there, Madison Becker. It's not transfer, but it's transcribe and translation. There you go. Transcription and translation. Okay, Sonia, what about the axon terminal and the sarcolemma of the muscle cell? What about that? You got a question mark after it. What do you want to ask me? these are structures. We haven't really talked about their functions yet, but they're a structure. Okay, remember, a function is something that occurs, but a structure is a substance, an item that's put together, and we look at how it's put together and try to understand it. Okay, that's what the synaptic cleft is, right? That cleft is the space between the axon terminal and the sarcolemma. It's a space. Remember, cleft is a space, like a cleft palate. Some kids are born with cleft palates. Okay, protein synthesis is what produces those proteins that you see, those purple little blocks. Remember, they're different from the actin, different from the myosin, but they play a role in what we call homeostasis and physiology. Now, out in that, let's look at that fluid for just a minute. That fluid that is between the axon terminal and the sarcolemma, that's extracellular fluid, ECF. So there is fluid between the axon terminal and the sarcolemma. Got some kind of connective tissue holding them in that position, but there's fluid in there. It's very small. It's got a lot of sodium in it. Okay. That's right. That's the other thing. Transcription and translation, Sonia. That's right. I wonder if this thing is sending these answers in unusual order. It's like they seem to come in a minute late or something like that. All right, let's start talking about the steps involved in muscle contraction. You know the connection with the nervous system and the muscle. You know the structure of the muscle fiber and the inside of it. So now we're going to talk about how does it contract. So steps involved in muscle contraction. Look on the next page, and you see basically the same illustration, don't you? Figure 1013. Yes. Sonia, space between, you're saying SC, or do you mean synaptic cleft? If that's what you mean, that's the space between the axon terminal and the sarcolemma. Thank you, Anna. 1015, if you're looking at the e-text. Did you get that, Destiny, since you're looking at the e-text? Good, got it, okay. So we're looking, in my book, it's 1013, and Anna Grace says for the e-text is 1015. There's a lot to be said for consistency, isn't there? You find your way through the forest. People are talking about the same thing. All right, so we're looking at either 1013 or 1015, depending on what you're looking at. And you see this neuromuscular junction, don't you? There is no figure. Well, there's a figure, Jason, but it's spread out over about 3 or 4 pages. What I'm going to get you to do is write it down sequence by sequence by sequence, and then you can learn that and then come back and look at these pictures. So let's look at figure 1013. And as Anna Grace says, it's 10:15. See the same connection. You got a neuromuscular junction. But look at the top of the axon terminal, and you see the green line? Notice to the right it says action potential. Another way to say that is a nerve impulse. Nerve impulse is a message through the nervous system. It's going to go somewhere and cause an effect. It's a change of ions, sodium and potassium ions. We're going to get to that in the next, not the next semester, the next meeting we have on Thursday. So we have the action potential or the nerve impulse. Like you want to pick up your pen or you move your arm and you pick it up. Send a message and it comes down as an electrochemical event. Doesn't make your legs jump. It goes where you tell it to. It's amazing, isn't it? So here you have the nerve impulse or the action potential coming down to the axon terminal. Second step, you see the ACH in the synaptic vesicle. Now, that vesicle is made of phospholipid bilayer. It's just like your cell membrane. That makes it very easy for that vesicle to fuse with the cell membrane of the neuron, and through exocytosis, you remember that? See, I told you stuff's coming back on you. You've got to go back and review it, make sure you understand it. Questions can come from there. But that vesicle with the acetylcholine in it fuses with the cell membrane, opens up, and out comes acetylcholine, the little diamonds. I quit scratching my nose, don't I? Now notice what those little diamonds do. They lock on to the proteins that pass through the membrane, the cell membrane. When they lock on, that opens the protein. And look what comes in. What comes through that protein? Somebody type it in. What comes through that protein? What is the substance? Sodium. That's correct, Destiny. That's right, Stephanie. Sodium. Now, why does it move in? That's good, Madison Oaks. Why does it move in? Because of a concentration gradient. You remember the word gradient? That's right, Mason. You remember the word gradient? It means difference, okay? So there's more sodium outside in the cleft fluid than there is inside the muscle. So the sodium, when the gates open, when the proteins open, in comes the sodium. Down its concentration gradient. That's correct, Sonia. Those two words are interchangeable. Now, we're not going to get into all the depolarization, okay? Well, you want to say it a little differently, Sonia. Sodium enters into the muscle cell. It's already there in the synapse between the axon and the sarcolemma, but it leaves that fluid and goes into the muscle cell. Chapter 3 dealt with exocytosis. We got about 5 minutes left here. So let's look on the next page. I'm looking at figure 1014 on page 354. Destiny, you found it yet? Figure name says excitation, contraction, coupling. Excitation, contraction, coupling. Tamika says it's 1016 in her book, 10.16. Got it? Okay. Now, as you look at this picture, you see there's your axon terminal to the top and the left. And you see the exocytosis, because that impulse has come down there and caused this event. And how in the world did that change and cause that event? We don't always have the understanding for that. We can sometimes look at something, but just observation of something does not mean we understand it. Why did it happen? Don't ever think science has got it all together. We don't. We know enough sometimes to help people, which is what you're going into in terms of the medical field, but we certainly don't have all the answers. So you look at that axon terminal, you see those synaptic vesicles dumping acetylcholine in. The acetylcholine connects to the proteins, which opens the door, and sodium rushes in. Now follow to the right those little positive charges. Follow to the right. Oh, and it takes a drop, doesn't it? This is where the T-tubule that we looked at. Do you remember looking at that? Let's go to, keep your fingers there in that page. And let's go to page 343. Keep your finger there. Go to 343, figure 10.4. So as you look on 10.4, you've got that picture. It's a greatly enlarged picture of a muscle cell. And you look at the top of that muscle cell. Now, don't look at the big muscle up there with the muscle fiber and the fascicle and the bone and all that. Look at the bigger one right in the middle. And you see sarcoplasmic reticulum. That's the purple kind of web-looking stuff. A little looks like it's got holes in it. There's actually tubes with spaces between the tubes. Then you have a transverse tubule. It's that kind of flesh-colored structure. And then you see to the right, it says terminal cisternae. And that's the inflated, enlarged area of the sarcoplasmic reticulum. So those are three structures now we're dealing with at this moment. Go to the bottom of that muscle cell where you see a little box on the bottom. And then you have that blue arrow that comes down to the bottom of the illustration, and you see the word triad. I think we talked about that, but if we haven't, we did now. So the triad is the two terminal cisternae and the T-tubule, the transverse tubule, which is just an invagination of the cell membrane. Now let's go back to page 10, or not page, but 1014. I'm not sure what. Let's see. Tamika says maybe 1015. I'm looking at 1014. Everybody back on that page we were looking at? We're just about out of time here. So you're looking back at the axon terminal. Exocytosis is occurring. That's going to cost energy. Okay. And then you see the little acetylcholine molecules attached to the purple proteins. It opens them up, and in comes sodium, and we follow the plus marks all the way down the T-tubule. And what happens as that charge goes down the T-tubule, it makes those two terminal cisternae release calcium. As that charge moves through the T-tubule, it causes the terminal cisternae to release calcium into the cytosol, into the cytoplasm. Everybody okay with that? So calcium is now in the cytosol. Look on the next page. In my case, figure 1015. I guess it'd be 1017 for some of you, maybe like you, Tamika, or whatever. Look at the little rectangle. Look at the little square. Actually, it's kind of a square, but it's more like a rectangle, though. But look at it down there, and you see the actin. That's the purple grapes. And you see the little yellow balls that are the troponin. And you see the blue lines, which is tropomyosin. Now remember, tropomyosin is blocking the active site of actin. So when calcium comes in, look to the right at the next rectangle, and you see the little troponin has two little calcium molecules bound to it. That's the little green ones. So calcium is released. It attaches to troponin. This connection, that's between the calcium and the troponin, This connection causes the tropomyosin to move, to change its shape a little bit. Causes the tropomyosin to move and expose the active site of actin. Okay, we're getting close to the end. We're going over 5 minutes. We can finish it up Thursday if you want to. You want to take a breather? Well, unless nobody's answering, let's go ahead and finish it. 357, page 357. Look at that long figure 1016 on page 357. Oh, Destiny, you got to go back to work. Okay, you want to stop all this now? All right, we'll stop. We'll pick up on page 357 and finish it up and finish this chapter up probably Thursday, okay? Any questions? Don't wait to study this stuff right before the test, unless you just want to make a zero. Okay, Cameron's going. She says, see you later. Mason says, no, sir, thank you. Okay, all right, we'll do it on Thursday. Keep yourself healthy, hands away from your face. Jason, I hope so, that the exam is next week. They should be telling us how to get this Honor Lock online. And then I'll send you maybe another test question so we can probably do it next week. Chapter 5 and 6, Kiara. Chapter 5 and 6. That's on the skin and it's on the bones. Don't forget to review chapter 3 and 4 a little bit, okay? All right. No more than 3 of you together now. Stay healthy, okay? I'm going to cut this off. See you folks later.